The method that we have published is one in which we use light to restore communication between two nerve cells. And this is important because in many diseases like Alzheimer's or Parkinson, there's a problem in communication between two nerve cells. And the classical ways of addressing these disorders is using drugs, and we showed like there's also the possibility of using light to restore this communication. And we have done so using the worm Cyanorhabditis elegans, which is an animal model very useful, because we know a lot of its neuronal communication. So we had to go through different steps to generate our method. The first was the choice of the defect that we wanted to create, between which nerve cells we wanted to create this defect. Then the second was the choice of the enzyme that we would use to generate the light. And this was also challenging because we needed it to emit light inside of the worm, but we also need to see this light. And we couldn't use the conventional microscopes. We had to design one at ICFO. Then we also needed to have specific ion channels in the postsynaptic cells that were very sensitive, so that very little amount of light would open them. And then finally putting everything together in the worm and make the experiments to see how the system would work in vivo. In previous experiments in which light was used to modulate the behavior, they had to use an external light source. And in our case, the light source is directly inside of the animal, because we have directed that particularly in the cell that we're interested in. With genetic engineering, we have coded for this enzyme, and we have introduced that in the worm. And this enzyme requires calcium and also the binding of a cofactor to emit light. And this calcium increase happens when the presynaptic cell activates. So it's very specific that this light is only on when the presynaptic neuron is on too. When this happens is when from the presynaptic cell we emit this blue light, and in the postsynaptic cell the ion channel senses this blue light and then activate the postsynaptic cell and transmit the information to downstream, to the downstream pathway.
Bioluminescence is basically the production of light within the living organism. The normal way that you can see light in a research lab is that you use fluorescence. You basically encode fluorescent protein within the animal or the organism, whatever you're going to image, and then you use a laser, an external source of light. This one will excite the fluorescent protein, and the protein will release some photons. In bioluminescent imaging, what you do is that instead of having this external source of light, you're going to place some a chemical. We call it a cofactor. So these molecules are going to go directly into the luminescent protein, and this will activate the enzyme or the protein to release some photons.
In our case, the cells do not need to be connected, they need to be close, because what we do is to shine light from the presynaptic cell. That's the first cell that receives the activation. We have broken this communication, and then in this cell that doesn't transmit the signal, we have coded for light-emitting enzymes. And these enzymes emit light when this neuron is active. And once this light is emitted in the postsynaptic cell, that's the one that is not receiving the signal because we have broken this transmission. In this postsynaptic cell, we express ion channels, and these ion channels are light sensitive, and they can sense the light that's emitted in the presynaptic cell, and so also activate the postsynaptic cell and proceed to the transmission of the signal.
So one of the main challenges about this study was to actually acquire the photons that were coming from the neurons, right? So it was very difficult to acquire this very dim signal. How we solved it was to build our own scope, our own microscope, to be specific for these experiments. In that sense, the sensor was very, very sensitive to just to a few photons, and you were able to acquire these very dim signals. We took the default model of a microscope, and then we start to remove the optical elements that you don't need for bioluminescent imaging. We just ended up with the sensor and the lens that it was going to focus on our sample. And after this, we just enclosed all these optical components in a box. Basically, this was just to remove all the light and to try to decrease the noise that you have in your imaging. So that's it. A very sensitive sensor and a lens to focus your sample. We also use artificial intelligence in order to enhance the bioluminescent imaging. So one of the critical things for an organism in order to survive is intercellular communication. This is critical because it gives you the timing and the functions that that organism needs in order to survive. For example, in the brain, neurons communicate with each other with chemical neurotransmitters. In that sense, with some diseases like Alzheimer, this communication between neurons is being disrupted. In this study, we demonstrated that these chemical neurotransmitters can be actually replaced with light. In that way, you can overcome this malfunction in the neural circuits in order to the neurons to communicate again.
Our method is an alternative to the classical therapies and methods that are used nowadays to restore communication or to treat diseases in which there is a failure in the communication. In this case, we use light. It's an alternative. Instead of using drugs or chemicals, we decided to use light, and we have seen that it works in vivo in a worm. So we think that it's possible to expand this. Of course, there's a lot of things to do, but it's like paving the way, for further developments and applications in the direction of neuronal communication, maybe in more complex cellular circuits or neuronal circuits.